Okay. DNA structure and function. All right, this is actually a more cooler thing. Now that we've established the basics of cells, we can talk about more complicated and more interesting things. Okay. All right. So DNA is a molecule. It's a type of nucleic acid. Okay. So I'm sure you guys know the basics of DNA already. Most cells use DNA to store genetic information. Okay, which is what cells need to do to produce protein. Okay, and I'm sure you guys know this already. Okay, this is Rosalind Franklin. Okay, and these two guys side here, that's Watson and Crick. These three people actually should get credit for determining the importance of DNA. That DNA is a double helix. Okay, so biologists determined the structure of DNA in 1955 by studying chemistry and using something called X-ray diffraction to see how the atoms are arranged. So basically, they shot a bunch of X-rays at it. Okay, and they realized, like, from this pattern that they got, that this must be how DNA actually works. Okay. Okay. DNA is composed of nucleotides. Okay, so this is a nucleotide. Okay. Okay, so the nucleotide has three parts. It has a phosphate group and a deoxyribose, right? This is D in DNA. And then it has a nitrogenous base. Okay, that's the nucleic acid. Okay, it has four possible nitrogenous bases. Okay, A's, G's, C's, and D's. Okay, these nucleotides form strands of DNA. Okay, and so you can form one strand here, you can form another strand here. Okay, these two strands wind together into a double helix. Okay, the two DNA strands are actually anti-parallel to each other. Okay, they're anti-parallel, and by anti-parallel, it means that one runs up and one runs down. Okay, they run in different directions. Okay, just exactly like I actually like this analogy. It runs exactly like a freeway. One runs north, one runs south. Okay. Okay, so the DNA strands are complementary to each other. So nitrogenous bases in one DNA strand forms hydrogen bonds with the nitrogenous bases in the other DNA strand, which holds the two nucleotides together. So A's always bind with T's, C's always bind with G's. Okay, and therefore, if you know one side of it, you'll automatically know the other side. Okay, so let's do this for example. Okay, so if my sequence is AGC, okay, then I know the other side. A's has to bind to T's, T's have to attach to A's, and the same for G's and C's, right? They have to opposing each other. Okay, so these have to attach to each other. So A's always pair to T's, T's always pair to A's, and then G's and C's. Okay. So if I know one side of the DNA sequence, I automatically know the other side. Okay. Okay, so again, if I know one side, I already know the other side. Okay, DNA is tightly packed into chromosomes. Okay, an organism's genome is all the genetic material in the cells. Okay, so all DNA is packed into chromosomes. Like DNA, uh, if it's completely unwound, it'd be too long to fit inside the cell and too floppy. And so we need to keep it organized. Okay, so DNA is wrapped around proteins. Okay, these proteins are called histones. The histones um, kind of bundle together into kind of telephone cord looking thingy, and then the telephone cord looking thingy wraps around the other, and that forms a chromosome. Okay, Zoom recording. Okay, so question number one. What's the main function of DNA? The main function of DNA is to encode proteins. Okay. Okay. So describe the components of DNA and its three-dimensional structure. So make sure you're able to do that because that will be on the quiz. Okay. Okay. So okay, so that's the structure, right? So two anti-parallel lines, both the nucleotides. Okay. And I will give you a DNA sequence, and you will have to give me the complementary sequence. Okay, that's definitely going to be on the test as well, but that's pretty easy. Okay, all right, next part. Okay, the central dogma. The central dogma explains how DNA encodes proteins. Okay, so the first process is called transcription. Okay, so transcription, you go from DNA to RNA. Okay, when you transcribe something, you're literally copying it over. Okay, so DNA and RNA are both nucleotides, so you're copying it over from DNA to RNA. Okay, in eukaryotic cells, this occurs in the nucleus. Okay, and then the RNA leaves the nucleus and is translated into cytoplasm. Okay, and that process is called translation because RNA is translated into protein. It's an entirely different language. Okay. So, make sure I'm recording. Okay, good. Okay, so a gene is a small region of a chromosome. Okay. The sequence of DNA of each gene encodes a specific protein. Okay. All right, so RNA, the protein production requires RNA. So the first thing we'll do is translate the DNA into RNA. So let me explain what RNA is. Okay, RNA is a nucleic acid that participates in protein synthesis. It does a lot more than that, but we'll keep it simple for now. Okay, so there are a couple of big differences. One, this hydrogen right there versus that OH right there. Okay, this is why this is DNA, deoxy. Okay, is missing that O, right? So ribose, you lose the O, you lose the oxygen, deoxyribose. Okay. Okay. So you have four bases: A, C, G's, and T's. Okay. In DNA, in RNA, there is no T. There is a U. Okay. So in RNA, A's pair with U's. Okay. U's pair with A's. Okay. Okay. DNA is double-stranded, RNA is generally single-stranded. Okay. DNA stores the information. Okay. RNA transfers the information and directly used to make proteins. It also catalyzes some simple reaction inside the cell. Okay. Okay. There are three types of RNA: messenger RNA, transfer RNA, and ribosomal RNA. Okay, and those are differences, but we'll talk more about them specifically when we actually do the process. Okay. Okay. So the gene is a recipe in the cookbook. So we like to think of our DNA as kind of like a big, huge cookbook where there's 20,000, 21,000 recipes. Okay. So there's actually 21,000 different proteins in our body. And okay, the cell uses DNA to synthesize RNA information, and then the RNA uses genetic information to synthesize proteins, and that process is called translation. Okay, so here the cookbook has probably a lot of stuff. Okay, and probably very big. You don't want to carry it around, so it's easier just to take a copy of a page. So you copy the brownie recipe, and then the brownie recipe tells you how to make it. Okay. Okay. So if a gene has a long nucleotide sequence, okay, the RNA will also have a long sequence. Okay. That does not mean you cannot have big proteins. That does not mean we'll have a genome. That does not mean there's no effect. Okay. But it means the RNA, if the DNA is wrong, the RNA will be wrong. Okay. Okay, so make sure you understand this too. Okay. Okay, so transcription uses DNA to create mRNA. So let's look at how it works. Okay, so the DNA is in A's, G's, C's, and T's. So A's bind with U's in RNA, C's bind with G's, G's bind with C's, and T's bind with A's. Okay, so if I give you a sequence of DNA, you should be able to tell me the sequence of RNA. Okay, so ACT, GAT. Okay, so A's bind with U's, G, A, right? And then the last three are C, U, A. Okay, so make sure you understand how to translate DNA to RNA. That will be on the homework. Okay, so, okay. Okay, so transcription builds mRNA, and this takes place in three separate steps: initiation, elongation, and termination. So initiation is where it starts. Okay, the first and last step are actually fairly difficult. The middle step is actually the easiest. Listen. Okay, so mRNA has to find out where to start. Okay, and there's an enzyme called RNA polymerase. Okay, RNA polymerase enzyme. Okay, will cause you to will find where to start, and it will start copying. You'll start making an RNA copy of the DNA. So you see that here in green. It matches the DNA, right? It copies over to the template. Okay, G's matches C's, A's matches U's, etc., all the way through. Okay, and that's the job of RNA polymerase. Okay, RNA polymerase unwinds the DNA double helix so we can get started. Okay, and then it copies the one strand of the DNA. Okay, and that strand is called the template strand. Okay, the other strand is called the non-template strand, and it's not using DNA synthesis. It's not using RNA synthesis. Okay. So it copies. Okay. And last but not least, it reaches the end of the gene. And in the end of the gene, there's a region called the terminator region. And it wobbles basically. You can see it wobble in that picture and it falls off. Okay. And then you're done. Okay. All right. In eukaryotes, the RNA is processed. Okay. So what happens, okay, is we need to add some processing to it. So let's start with what the processing is. Okay. First of all, RNA processing attaches a cap and a tail. Okay. So you have a 5' cap and something called a poly A tail. Okay. Second thing, RNA processing removes introns. Okay, so what happens is RNA has sections called exons. Okay, and regions called introns. The introns are then cut out of the DNA, and all you get left is that. Okay. Okay. So the processed RNA is now a functional molecule. When processing is complete, it leaves the nucleus, so bind to a ribosome, so translation can begin. Okay, so make sure you know how to do this. Okay. Okay, make sure you know how to answer this question too. Okay. Okay. This is translation. Translation. Okay. The cells translate the mRNA message in a sequence of amino acids. So here's a sequence of amino acids. Okay, and then in translation, this is translation, right? So here. Okay. Okay. So all cells have the same genetic code. Okay. The code is formed in 3-letter blocks. Each 3-letter block is called a codon. Okay. Okay. So the codon chart shows the genetic code. Okay. So the next thing you guys have to learn is how to go from mRNA to amino acids. Okay. So I will give you on the quiz, I will give you this sequence. I'll give you this chart. So do not spend your time memorizing this chart, but you have to know how to use this chart. Okay. Okay. The tRNA translates, so this is what tRNA looks like. It translates the mRNA into amino acid. Okay. Each tRNA carries an amino acid that matches the codon. Okay. All right. Translation. Okay. Require ribosomes. So this is what ribosomes are actually for. Ribosomes are simply where place where all this translation actually occurs. Okay. Okay, so all 3 types of RNA actually come in to make the ribosome work. Okay, the messenger RNA is the message from the DNA. Okay, the tRNA, I already said, each tRNA codes for an individual amino acid, and the ribosomes themselves are made from ribosomal RNA. Okay. So, right. So, okay, I am recording. Okay, so here, the ribosomal subunits are actually made from RNA, the mRNA, and this is the tRNA. Okay, translation goes to protein, same 3 steps: initiation, where it starts, okay, elongation, how it continues, and lastly, termination, okay, how it falls off. Okay. Okay, initiation. Okay, mRNA is a template. Okay, all right. Okay, the ribosome holds it all together. Okay, so tRNA, amino acids join, blah, blah, okay, until the very end. Okay, so what actually happens is the very first thing that happens, okay, is initiation. It's initiation. It takes the mRNA sequence and looks for the very first AUG. Okay, that's where it knows how to start. It takes the whole mRNA sequence, looks for the very first AUG. Okay, okay. The AUG will bind to tRNA, the first amino acid, okay, and the ribosome will clamp on. Okay, once the ribosome clamps on, it invites the second amino acid to join. Okay, the first AUG, the first amino acid has to be AUG. The second one to the very last one doesn't have to be AUG, but the first one does. Okay, so here's the first AUG. Okay, it brings in the next amino acid. Okay, here, so the next tRNA. So the CCU matches the GA here, and you have amino acid here, glycine. The methionine and glycine, since they're so close together, can bind together and form a peptide bond, and then everything shifts to the right one, or in this case, the left one, and then you do it again, and again, and again, and again, until you reach a stop codon. Okay, so you always start from methionine until you reach the first stop codon. Okay, in class on Monday, I will make sure you guys understand this process. Okay. Okay. Translation is efficient. Okay. Multiple ribosomes can actually attach to one mRNA molecule. Okay, so you can see the ribosome starts from this side, goes to this side. So as it goes, okay, once one ribosome starts forward, another one can clamp on and move through, and then another one can clamp on. So you can constantly make these proteins. So you can see these are shorter, these are longer. So these must be first, and these must be last. Okay, so make sure you know how to do this. Okay. Okay. So cells express their genes. Okay. So all cells have ways to control the rate and timing of transcription when the genes are made. Okay. Eukaryotic gene cells often control the rate and timing of translation. Okay. So Okay, I'm gonna stop this presentation here. It'll be part 2.